just started, finished up the issue of recording the meetings, the presentations, really what it's about, and getting it posted onto, actually, I think a couple of sites are Facebook and somewhere else. And so I'm not sure if that, because we've never had like under 15 people anyway. So I don't know what has happened, but anyway, start the business. The presenter is Paul Arnold. I'll give him the full introduction in a little bit after we get through the business. The first thing on the agenda is that in June, for our June meeting, Greg Beyer, back there, our videographer, is going to be occupied with the Scottish Highland Festival.
Where'sville Scottish Festival and Highland Games.
Oh, it's two different things?
No, it's one thing.
It's one thing.
Long,
Okay.
one long name.
With
One long thing, okay.
Yeah.
And actually, our June meeting is going to be in here. I mean, in the Civic Center, not in here, because they're apparently having book sales here. But it's going to be in the Civic Center. Next month we're going to be here. So if anybody has like any sort of filming skills or anything like that, Mark, do you have filming skills or anything? Okay.
I can do it, Teryn. He's just recording it to a memory card, so it's more or less set up the camera and turn it on and point and shoot.
Yeah.
Okay.
Zoom in and out to get your frame and then hit record.
We don't want to miss filming now that we've got it started, but that may be part of why there's so few people here. Thank you, incidentally, therefore, for anybody that is here. Okay, our new members, Kim's the one that is responsible for that. Jacqueline Schultzke, Deborah Smith, Jonna Tuttle, who was previously a member, then had to sort of back out with other stuff she's doing, but she's back in and very involved, and Ann White. And I'm presuming none of those are the people that are here. Welcome to them. Wendy Beyer was here. The videographer's wife is our treasurer. And so she had to leave to do stuff also. But she said, finance, no change. Okay, so that's it. We haven't spent anything. We didn't have anything coming in. We're good. Okay. Projects that we have going on, the Beasley Knob Native Plant Garden. 2 weeks ago, we were supposed to be working on doing the post-winter maintenance, weeding it out, planting some additional plants that came from the botanical garden. But we weren't able to do our work then because it turned out that it's on state forestry property, and they had donated the fence that's going to outline the bed, the Connect to Protect garden. And it turned out that's when they were going to install the fence. So we couldn't be doing that now. So then it was postponed till this past Thursday, which had heavy rains and winds, and so it wasn't done there. So it's going to be next Thursday, we hope, on the 18th, which now we have conflicts, but hopefully there'll be enough people there to do their work on that. Our plant sale
That is from 11 to 1.
From 11 to 1, yes. Previously we had done 10 to noon, had been what had been scheduled, but 11 to 1, thank you. Okay. Plant sale in June, this is our main income generator. I don't imagine many of you are going to want these pots. I was just bringing these to donate to anybody that's planting things, particularly the upsize, the pot, if you're transplanting some shrubs and stuff, we need them, and here you don't have to go buy the pots. But if you need any of those, feel free to take those. And I also brought some samples of plants. It was Jonathan, was that in
January.
January, he did a presentation about the arrow gardens. Little way to get seeds going. We bought 2, and these came from there. So that is a very effective way to get plants started. There's ones that I was doing just from seeds into little pea pellets, nowhere near where these guys are. These 2, the dwarf-crested iris and cinquefoil, are ones that I've transplanted from my property, from our property. that's just sort of to let you see how wonderful the arrow garden thing is. And it also sort of shows that you need to have a little name tag for each thing, identifying it, so that once we get to the sale, people don't have to say, I want this, but I have no idea what it is. We know they're all natives. Okay? I also have brought some flyers, I don't know where they are here, but if you have a place where they're printed out, not like little ones to hand out, but to post somewhere about the plant sale. I'll see if I can remember where I put them. They're in one of those boxes. It's not a big deal, but before you leave, if you feel a place to post that to help promote the sale. Okay, recent events. We had a board meeting by Zoom this past Monday, which may not seem very, very interesting. Thank you. Good. There we are. But we're supposed to have quarterly board meetings, and we did have one in like March of 22. So anyway, we're trying to get back into that. And some of the things that we discussed were the field trips, which we have pretty much lined out for the summer, trying to get some garden club contacts, and other ways to get information out, how we can connect with municipalities to try to get their contractors to use native plants and things like that. That may depend on the club growing a little bit more before we have enough people to do that. Okay, upcoming events. The Sati Nakuchi native plant sale is on the 20th, which is like just next week, I guess, Saturday of next week. And there's information there on the internet. There is at Lenfoot Hike In, which is over towards, where is that? It's near the beginning of the Appalachian Trail, so it's near there. And they had connected with us because they had native plant gardens, but they haven't been controlled or maintained in a long time. So what they're doing is getting that reestablished. So they have some volunteers that are going to be doing that, which is much appreciated because it will promote native plants. That's cool. And there's Homegrown National Park is the Doug Tallamy program thing, trying to say, everybody just makes their property a homegrown, native plants and promoting pollinators and reducing the grass area, the sod type grass. That the more we have of that, the better it is for the whole nation. And there is a challenge from April 1st to July 31st to try to, you don't have to have your whole property go native. Just do a little bed, and then you can put it on the internet, on the national park, the homegrownnationalpark.org, and it gets represented. People can see what states are doing better than what other states on that. Okay, any questions about this stuff, the businessy stuff? I don't print out a lot of agendas, but there's enough that you can each have.
Did you say natural park or national?
National Park. It's called info@homegrownnationalpark.org. So you can go ahead and pick up the agenda there and have it segment. Okay. Introduction. Dr. Arnold back here, Paul Arnold, has an AA in field biotechnology. It seems like an AA, you wouldn't think it would be that refined at that point, but BS in biology. BS is not bullshit. It's BS in biology. PhD in botany from Miami University in Oxford, Ohio. He's now at Young Harris College, so when we were meeting there, we see him frequently. Teaching plant physiology, beekeeping. So is that actually a college course, is the
Yep.
beekeeping thing?
I teach
So
as
good.
well.
Okay,
It's,
nice,
they get it for credit,
good.
and they play with bees.
And they don't get stung too often, right?
Not too often. I get stung more than they do.
General biology, ethnobotany, and mycology. You know what mycology is? Mushrooms, fungi. Fungi. His research includes plants, insect relationships related to forest health and apiculture, bug stuff. Plant and mycorrhizal relationships in the Appalachian plant diversity. He oversees Young Harris College Hemlock Project, which uses biopredatory beetles to control the Hemlock Wooly Adelgid infestation, and enjoys working with the students, which I miss. I was a high school teacher, but still, as they explore the natural world. His other interests include hiking, geocaching, okay, where you're looking for things that are hidden, music, and sports. So here he is, and hopefully everything's lined up.
Glad to be here. Thank you, Teryn, for introduction and for allowing me to talk to you.
Hold this while I pull this out.
Oh, yeah.
One of the things that we do is, as Teryn mentioned. By the way, field biotechnology is a real fancy name for wildlife management.
Okay.
That was actually the name of the degree when I started the program, and when they decided to change the name of the program kind of at the end. And I always thought that's one of the most ridiculous names, when in reality wildlife management really tells exactly what I was doing back then. But and my claim to fame in wildlife management was probably planting acres and acres and acres of autumn olive, which is pretty terrible. But back then, in the 70s, autumn olive was frequently planted as for wildlife habitat and other things like that. So I have single-handedly probably messed up. So that's unfortunate, and I pay penance for that every day, I feel. So I'm upfront about that faux pas in my past. So anyway, what I want to talk to you a little bit about is Hemlock Wooly Adelgid mitigation. And I think you all have probably heard of Hemlock Wooly Adelgid and the hemlock trees. I am going to talk a little bit about hemlock trees, and, just because, I love them so much, and so I'm going to mention a few things about them. But I'm going to try to focus on some of the things that we're trying to do to keep the hemlocks going in the southern Appalachian area. This area has been hit very, very hard by this pest, and so I'm going to talk a little bit more about the pest itself. And in fact, there's some things about the pest that decide how we approach it, and I want to talk a bit about some of that. So, probably most of you are familiar in a Georgia Native Plant Society meeting of eastern hemlock. Eastern hemlock is our main hemlock that we have here in the East Coast. Interesting thing about eastern hemlock and Carolina hemlock that I'm also going to talk about a little bit is that those trees are kind of hemlock outliers. The rest of the world has hemlock species. There's hemlock species in Europe, hemlock species in Asia, there's hemlock species on the West Coast, and all of those hemlock species have been exposed to Hemlock Wooly Adelgid probably for millions of years. Even originally, we used to think that the West Coast trees, that the Hemlock Wooly Adelgid was introduced like in the 20s. Well, we now know through genetic studies that was not the case. That has been actually a part of the hemlocks from the West Coast for ages. And so, probably for millions of years. So, the reason I'm mentioning that is that almost all other hemlocks around the world have a modicum of resistance against Hemlock Wooly Adelgid because they evolved with them for such a very, very long time. And however, the hemlocks here in the east were kind of in a sheltered area. You had the Great Plains on the west, and plus before the Great Plains we had a Great Sea out west that kind of protected or isolated this particular hemlock group. And so the eastern hemlock and the Carolina hemlock never really were exposed Hemlock Wooly Adelgid. That's what made this so devastating. So when this was introduced in the 1950s into the East Coast area, it had a terrible effect on hemlocks because they don't really have any natural resistance, and we don't have many native bugs that really do anything or do much because they've not seen the Hemlock Wooly Adelgid as well. Now there are some bugs that help, but most of them are not actually non-natives that were introduced previously, like harmonia beetles, which are the regular ladybird beetle or ladybug that was introduced in great quantities, around the 50s and things like that, to help aphid control. So, well, this is a magnificent tree. I don't know if you've ever seen big hemlocks, but they're unbelievable. So they can grow up to 160 feet in height. Some of the ones in Joyce Kilmer, before they died of this disease, were that tall. Sometimes as much as 7 feet in diameter. So if you can imagine, it's almost like ancient chestnuts, where before the chestnut blight came in. You can see this kind of a recurring story of exotic species coming in and causing problems. And then old hemlocks, there were some in the Joyce Kilmer area in North Carolina that were found to be many hundreds of years old. I think the oldest ones that have ever been found have been 800 to 900 years of age. And in some cases, the Hemlock Wooly Adelgid came in and killed them in 3-4 years. So that's how devastating this disease is. So you can see why I'm kind of passionate about this. I'm a big tree person anyway. I've always been interested in forestry and especially forest health. I didn't want to be a person doctor. I like being a tree or plant doctor. They don't scream as much. Obviously, they are not only pretty amazing trees just in their own right, but they are very important for riparian habitats or river-type habitats. They are very prominent in North Georgia mountains in that type of an environment. And that actually helps do things like lower water temperatures and other things like that allows, our native brookies and other kinds of fish and aquatic invertebrates and other things like that to thrive. Without the hemlocks, you lose a little bit of that shading of creek environments, which can actually upset the intervertebrate mix and the fish, the fish reproductive cycles. So these trees do a lot of good. And of course, so do rhododendrons and things like that tend to be along shorelines too, but they're part of the mix. A lot of bird species feed on their seeds or nest in them. So you have, these are trees that contribute a lot to the ecosystem. And yes, we're people, we deal with money, so I need to talk a little bit about the economics of the situation. They, of course, from just our eyes, they're aesthetically beautiful. And they are often used as a landscape tree because they are so beautiful. And of course, a lot of times, especially in the beginning of this, the biggest worry about Hemlock Wooly Adelgid was economic worries, because people would often have acres of hemlocks on their property that made it look like a tremendous, beautiful forest. And they, of course, were worried about losing them in terms of property values, in addition to, it all ties together, of course. I mean, it's all part of the package. So this is actually a picture I took of one of the trees, very close up, trees on the college campus. I don't know if you've been at Young Harris College campus, hemlock trees are very prominent. We have 3 very old hemlock trees that are over 100 years old, kind of in the area of the chapel buildings, by the way, the bricks were made by students back in those days.
Wow.
And so, you know, so historically, there's some pretty amazing places. But anyway, very small cones. I know these look gigantic, but they're about this big. And of course, in the case of the eastern hemlock, the big notable way to tell, or one of the big notable ways of telling the eastern hemlock from the Carolina hemlock, which is also native, not necessarily here in Georgia, but some of the higher elevation regions in North Carolina. But the eastern hemlock tends to have persisting cones that are brown that stay on the tree even long after they release the seeds. And the cones, kind of dangle down. And so and they tend to have their leaves come out in what we call it two-ranked, which basically means they kind of describe like a flat geometry on the branch. So that's kind of how they look. That's probably not totally evident in this picture, but one I'm particularly worried about, the second species that lives in the eastern U.S. is the Carolina hemlock. And this is a picture, oh gosh, I think it's up around Highlands. I forget exactly where I took this one. All I remember is I was climbing around a lot of rocks when I took that. But, or actually, no, this was Robert Jedden. Yeah, he was with me. So he's the one who took the picture. So I was just kind of tagging along, just trying to stay alive while we were making our way through these regions. But, Carolina hemlocks are more shrubby. They don't grow tall. They don't grow large. They're basically higher altitude hemlock trees that have adapted to pretty rough environments. And they also have a difference in their branches. Their needles kind of come out in all directions, not just in that two-rank. Their needles are longer. And also, they don't make brown cones. Their cones are more like yellow, and they fall off the tree immediately after they release their seeds. So there's a few ways that you can tell these different species apart. The biggest thing is that these are very rare, and they also are very susceptible to HWA, Hemlock Wooly Adelgid. And so that's the big worry. This tree is probably a good candidate for listing because of HWA. So, unfortunately, it's maybe even more susceptible to it and affected by it than even the eastern hemlocks are. So, the villain. The villain is HWA, otherwise known to us as Adelgid tsugi. And this particular bug was introduced in the Richmond, Virginia area around 1952. It was imported on oriental hemlock trees used for landscaping, and mainly Japanese hemlock trees. And unfortunately, the Japanese hemlock trees do have resistance against this bug, so they can carry it, they can have it without suffering severe consequences. And then the other fortunate thing is it took a long time for this to get spread from Richmond, Virginia. Richmond, Virginia is not in hemlock country. The only trees, it was just kind of an oddity that this disease existed down there with some folks that had landscape hemlock trees in their yard, and then they got this disease. And so that was how it kind of drew our attention. But everybody was a little afraid that all it would take is somebody to drive from Richmond, Virginia, take a trip along the Blue Ridge Mountain Parkway or something like that, and then boom. And that's probably what happened. These bugs, they are very, very tiny. They're probably about like a half to a quarter the size of a period on a regular written piece of paper. So they are very tiny bugs. They pierce the base of the leaves. They tend to suck on the plant juices. And in addition to that, their saliva has chemicals in it that make, that harden the existing tissues. So once they are on a tree, they, their saliva actually contributes to the problem. It tends to block up a lot of the phloem of the tree, which is the conductive tissue of the tree that conducts carbohydrates and things like that. So that means that the roots and other parts of the tree don't get the carbs that they need if there's a horribly infested tree. Now you might wonder, well, why would a little tiny thing like this be a problem? Well, part of it is that their mouth part that they introduce into the tree is well over 20 times the length of their body. So they can extend it and they can go into cells all over the place around them. The other problem is that there are so dang many of them. They reproduce. I'm going to talk about that reproduction in a second. So usually you'll see damage within 1 to 2 years of it coming on the tree. You start to see drying out needles. Sometimes we call trees with these desiccated needles gray ghosts. I think I've got a picture of a gray ghost on here. And then they start dropping their needles, and sometimes the death of the tree can happen as early as 3 years after infestation. Probably more likely at 9 years. And there's some trees that hang on longer, but typically trees don't tend to survive unless there's some kind of mitigation on this particular bug. Now here's the big problem. Adelgids are parthenogenetic, very similar to aphids. And what that means is they produce eggs that don't need fertilization and will produce more females. So just like an aphid can feed on your plant and then can pop an egg out, and then another female aphid will feed on your plant and it'll pop an egg out, and then another female aphid. And, just like with aphids, adelgids, although they're not in the same group as aphids, they are somewhat similar in their life cycle. Their life cycle is a lot more complicated than an aphid, but still they do that, which means that every female adelgid can produce offspring. You don't need males. In fact, here in the United States, males are not really a part of the picture. They're produced, but they fly into oblivion. They don't participate in sexual reproduction in the U.S. because they have a second host tree, an oriental spruce, Asian spruce, that they actually have to do their sexual cycle on. So luckily we don't have those here, so they don't do the sexual cycle, but they're pretty darn good at reproducing without that. So they hatch from unfertilized eggs. So basically here in the U.S. they're all females. They have 2 reproductive cycles a year. Each female can lay up to 300 eggs in its lifetime. Doesn't need a mate to do it. And then there is a sexual stage. It's kind of a winged adult stage. They fly to those Asian spruce trees, but here in the U.S. there's no place for them to sexually reproduce. Thank heavens, because sexual reproduction mixes the genetics up a lot more, and that increases the likelihood that anything you're throwing at them, they'll become resistant against. The good thing about not having a sexual cycle is their genetics remains a bit more constant, although they probably have a pretty good mutation rate, which means maybe it doesn't matter anyway. But they can, with the asexual stage, that's their fastest mode of reproduction. So typically there's a stage called the crawler stage. This is the stage when they are the most problem. That's what the stage that they spread. They don't fly, except for that sexual stage, which is pretty meaningless around here. But all of these little female adelgids, their crawler stage is able to crawl everywhere. And of course, you might have millions of them on a tree. So a bird lands on a tree, a couple of crawlers get on their legs, they fly tree and voila, you got it spread. You park your car under a hemlock, you got hundreds of thousands of crawlers crawling in your car, and you drive somewhere else, you do some trail hiking where there's hemlocks, and I think you all get the picture. It just can spread like crazy. We see them, they're noticeable to us through their very white, fluffy egg cases. So that's usually how we know we've got the disease. So this will life cycle the Adelgid, and the reason I'm showing this to you is because for our purposes of using other bugs to combat these bugs, it's important to realize that one of our goals is to actually attack it on many fronts, on different parts of the life cycle. So typically, now these days or months are actually a little off because this is information that came from the New England area. A lot of the original researchers on the Adelgid were from University of Connecticut or the USDA lab and the Alampo lab in New Jersey. And that's where most of this original research happened. We found that once they came down here into Georgia, we kind of had to throw away a lot of our knowledge of the actual life cycle in terms of the timing. So, I mean, it's the same life cycles, it's just that the timing's a little different around here. So we have two phases. We have the cistern stage. This is the generation that overwinters, and then we have the spring generation, which is the progredience phase. So right now, we are pretty close to what this says for the progredience stage, but we still have progrediens eggs that are hatching right now among the Adelgid, and we're producing these different stages of the Adelgid that will eventually result in adult females that will then secrete this wool around its body, and it lays its eggs basically on that wool around its body. So it produces its egg sacs. The cistern stage, around July and August and September, you'll notice there's not a lot of change, and that's because we call these the cistern nymphs here, and these nymphs are basically dormant. They just are not doing anything during that period of time. And so this is a hard stage to attack because these nymphs are very, very tough on the outside of their body. They're very hard for other insects to eat and attack. And so a lot of the predators that we rear don't necessarily like them. They just don't like the taste of them. But then we have, as we get into the fall, molting a few times, creating different stages till you get an adult. And then typically around January, which is when we really crank up our lab to try to rear these beetles, then we have these adults. These adults are then pretty dramatically going into the hemlock trees, attaching to them in numbers of millions, and then sucking the juices out of the tree. And then there's the female cisterns stage with the egg sac around it, and then it'll lay its eggs, and then we go through this again. So it's a little off in terms of months here down in the south, but still, generally it's the same type of life cycle, just the timing's different. Well, anyway, that was a female Adelgid.
Not me.
Like there was a surge.
It might have been.
It looked like a surge, because
Yeah.
the lights flickered too.
We're still running.
Still on, okay.
Okay. Turn the projector back on. Yeah.
Or a little hot or something. The lights kind of.
But anyway, that was, you saw for a split second, this kind of very branches from the field. We basically have to feed our bugs with these branches and hemlocks on the branches. So that's kind of how we do that. So this is that burnt popcorn kernel right there. This is the cistern's nymph, and it's just highly. We sometimes use the word sclerotized. In other words, its exoskeleton is just very, very tough.
Okay.
And it is basically mainly just waiting out the summer. The word estivation means, it's like hibernation. Hibern means winter, and esta means summer. So, so that's why it's like a summer.
So it's not actually damaging the tree when it's at this stage?
No, at this stage, it's just kind of hanging on, chilling, I guess you could say. Yeah. So, a lot of these photographs, by the way, Michael Montgomery, amazing scientist in the area up around Virginia Tech, and he did a lot of the groundwork and understanding the life cycle of the Adelgid. So a lot of his work really helped the rest of us to jump in and try to figure out what we could do. This is one of our photos, and this is another way of looking at it. This is a hemlock needle, so you can kind of see what the size is.
Wow.
This is the hemlock needles are actually pretty small, but there you can see it. And so at this stage, we are now beginning, those nymphs have become cistern's adults. And in this case, they are, you can actually see the wax that they're secreting to begin their egg sac. So this is right at the beginning when they're about ready to start laying their eggs. And then these are the egg cases. This is usually what you find on your tree, and you see those. Kind of looks like it snowed, and
On the underside of the.
right on the underside of the branches. So this is pretty devastatingly dense infestation here. Each of those little wads of cotton are actually little waxy egg sacs from a single female, and there could be dozens and dozens and dozens of eggs in each one of those. The Adelgid crawlers, that's how big they are. There's a penny for comparison, so you can see that. It's funny when I look at them, if I like to have a white sheet of paper on the lab table when I'm working with these, you see these little, very tiny dots just moving around. Those are the crawlers. And that's the stage where they spread. This is the most current map I've been able to find, the spread. I think the Forest Service is working on an updated version. This was from 2022. You can see that the Appalachians are basically totally involved with the HWA. It actually started more in the northern aspect, and then it kind of came into Georgia probably around 2002, around the High Shoals area, or Chattooga River area. That's probably where the oldest infestations are in the state. And now you can see it spread through basically all of the hemlocks in this area. This to me is a little dubious. This map says that there's none found in, I guess that's Cherokee County. I don't know about that. I've had a few people call me from that area saying that they've seen it. So, and then there's a few hemlocks in northwest Alabama that appear, areas that appear to have escaped so far. But basically, it's through the whole region. You're probably wondering, well, why all of these hemlocks up here are not infested? Yeah, they have something called winter up there. I don't know if you've ever seen it, but we don't tend to experience it much. But winter really does knock down the Adelgid population, and so it helps prevent their spread. So a good winter is an ally for us. And so when we have these really cold spells, that sometimes knocks down even our populations. So So I personally would like to see a bit harder winters to combat this disease. Just very recently, it's now in western Michigan, and this is of course the area that I'm from, that area. And so up in Michigan, just magnificent hemlock stands. So it just kind of hits me there that isn't infested up there. Of course, much of Ontario. So far, a lot of areas untouched. In Georgia, this, of course, the Georgia Forestry Commission has come up with this map. There, you can see how it spread, beginning at around 2003. I actually am pretty sure we had it in 2002 and maybe even 2001 in places like High Shoals and the Chattooga River area. It's hard for me to think that when I first saw infested trees there, it was already 2003 and they were already heavily involved. It's hard for me to think that that would have happened just in 1 year. So, and then of course towns in Raven County and northern White County. And it stayed here for a couple of years and then it kind of exploded out into the Cohuttas and into the Rock Creek area and all of that. All of the areas kind of west and south of here. And then, of course, now it is involved everywhere. It has involved Chattooga County, Walker County, Whitfield County, Gordon County. Hemlocks are infested there. So, the places around Cloudland Canyon and that area, the Lula Land Trust, which is in that region too, highly infested. We've done a lot of beetle releases in those areas the past few years. We tried to release as much as we could on the front lines to try to knock the populations down as fast as we could. And hopefully that's worked a little bit better than some of our strategies in the past. So, bad news, yes. A lot of loss in the Appalachian Range. There's some places, including here in Georgia, where 50% of the stands have gone. And more so even in places like Shenandoah's, the Shenandoah area, it's probably more like 80-90% loss. And it happened fairly early on in the infestation when it came. Of course, you lose that stream shading, which can affect the ecosystem of the stream, the birds that depend on the tree, many other things. You lose that aesthetic beauty. We unfortunately don't really have native natural enemies of this particular bug that we know of. One of the new experiments that we're doing in the lab is we're actually trying to see if there are some natives. And there's kind of a technique that's been developed, and I'm going to talk about it now. We try to do that. So this is what a gray ghost hemlock looks like. A hemlock that's been infested, instead of those kind of nice bright green needles, it's more like kind of a gray look. And so like I said, we call those gray ghosts. They, when we see them in the field, we know we've got an advanced infestation.
And is that too far gone to inject the poisons around them to try to save it, or is it just too far gone?
It might be too far gone. However, probably use of safari, if anything, will bring it back. It's the compound that is known as safari, or dinotephrine is its actual name. Safari is the brand name. Safari has very quick uptake into the tree. And so sometimes I've seen trees like this come back with that type of treatment. Imidacloprid, which is the main treatment we use chemically, I think that would be too late.
Yeah, that's the slower acting, longer lasting.
Yes. Yeah. So speaking of the devil, we do have chemical treatments, and I think you've all had Donna Shearer come and speak to you all, I think maybe last year, a couple of years ago. So she was the one, she and her husband were the folks that kind of organized Save Georgia's Hemlocks. which has a website. Everything you want to know about hemlock infestation in the U.S., you can find there. And there's some little flyers there that you had shown me that you got there. So if you, I guess feel free to pick them up.
Absolutely.
It's a wonderful resource. Donna's had to back out a lot. She's been involved with it a long time, but there's other folks that are kind of carrying the torch for. And that group has also organized volunteer groups to chemically treat infested hemlocks in conjunction with the Forest Service and other groups under the supervision of other groups. So anybody who's really interested in doing that kind of thing, call St. George's Hemlocks. They have a hemlock hotline. So if you got hemlocks to your house and you wonder what you want to do, go and call that number, and somebody will answer that knows about hemlocks, and we'll be maybe talking through different options. Their website also is very good at that if you don't want to talk to people. I kind of like to talk to people, so I like to actually have phone conversations. Most of my students do not, so so if having a phone conversation or some of my students is sometimes hard. So among chemical treatments, of course, there's imidacloprid. That's kind of was the first in the arsenal that was developed. The imidacloprid's been used for years for aphid control and other things. There's tree and shrub insect control that's been around for quite a long time. It's basically an imidacloprid solution, and it can be used to treat the hemlocks. Big thing about imidacloprid is that it is slow to get into the tree, and it persists a while in the tree, which is good news for the tree. It could be bad news for persistence in the environment if it's applied into the soils. But it is a neonicotinoid, and these chemicals are, many countries have investigated them, and some have even banned them. We still have them in the U.S. And if applied properly, these probably are going to be probably the best solution for a landowner that has a few trees. And you usually will apply it into the soil environment around the tree. Remember that the feeder roots of a hemlock, in fact all conifers, feeder roots are right by the trunk. Unlike all other trees like oaks and maples, where the feeder roots are along the drip line on the outside. So if you were to try to feed your trees big hardwoods, then you wouldn't go near the trunk because the feeder roots aren't there. But conifers are designed to draw the water down the trunk, and their feeder roots are right there. So you usually will put the formulations of imidacloprid right around the trunk. And there's formulas that you use. Those formulas are actually on the website, St. George's Hemlocks, if you wanted to do it yourself. We now have pellet formulations called Cortec, which is which is a lot easier to apply than using the old soil injectors or drenchers. So, but it takes about 18 months for this chemical to get the peak concentration in the tree, according to most of the research that's been done. So that's why I said, with like the gray ghosts, it is 18 months may be too long to wait. So, second formulation is dimetephrine. We call it Safari. Very mobile, which is once again good and bad. The good part of it is it gets in the tree quickly. Another good part of it is that you can bark apply it. You don't even have to touch the soil, and it systemically will go into the tree. And it reaches peak very quickly, and hence it can knock down the adelgid fairly quickly. And sometimes there's a 1-2 punch that people use. They might just to keep the tree alive, they'll do the first punch with the safari, and then the second punch with the imidacloprid for long-term effect. Imidacloprid, by the way, once a tree is treated with it, sometimes that tree doesn't need to be treated again for 7 or more years. The older the tree, probably the more you would have to treat it. But 7 years, very commonly, you can go on one treatment. Dinotephrin's not that way. It builds up quickly and then falls quickly. And so it's very short-lived in the tree compared to this. If you want to go more the organic way, you have horticultural oils or insecticidal soaps. They do work. The problem is, if you've got eggs on there, it won't work on the eggs. It will only work on the adults that are active. And so that means that you're going to find yourself drenching that tree with the oils very, very common, or a lot. So it used to be how we would apply the chemical like imidacloprid to the tree through what we call soil injection. These things have become very hard to find, soil injectors. The company that made almost all of the soil injectors in the world was called Kairos. That company stopped manufacturing them. And so even St. George's Hemlocks, they basically have people in that group that one of the only things that they do is repair soil injectors to try to get them to continue to be functional. But you don't really need it. With the Cortec type capsule or the solid tablets, you can actually drill those into the ground around the tree, and that does probably just as good as the injector would. So that's soil injection. Some folks would do stem injections where they would actually drill into the trunk of the tree. Don't ever do that. It was found that it just doesn't work with imidacloprid. It might work with safari, but why do it with safari when all you need to do is paint it on the trunk and it'll go into the tree? So, the final thing I want to talk about is what do I do? And what do my students do? We have been interested since since the Adelgid came into Georgia. We've been interested in trying to find alternative ways to chemical treatment. There's problems with chemical treatment. Chemicals are, expensive, so you have an economic problem. If you were to buy enough chemical to treat hundreds of thousands of acres of national forest and all of those hemlock trees, it's prohibitive. Not only is it prohibitive economically, but ecologically. You don't want to have all of that chemical in the environment. It's probably okay if you've got a few trees on your property, or even selecting certain trees that you want to preserve even in a forest environment. But if you want widespread control of this, chemicals are always going to be a short-term and imperfect solution. And, so there may be another imperfect solution. And that's the one that we've been working on. So in our lab, we are rearing 2 species of beetles. At one time we were rearing 3 species of beetles, but the third species, it turned out it wasn't as selective in its eating of the adelgid, and so we contacted Rusty Ray's office, who's our forest health person in the Forest Service, and said, I don't know about these anymore. And then he said, yeah, we're going to stop that. So that third species, which we reared for a couple of years, we decided to stop because it wasn't just eating the adelgid. The interesting thing about both of these species, Laricobius nigrinus and Sasagiskivus tsugi, and by the way, let's just call them LN and ST, just for sanity's sake. Laricobius nigrinus show you a list of those labs. So, Laricopius is actually a native to the US or Canada. So in that sense, it makes it an easier bug to work with because you don't have to worry as much about quarantine controls, although we still had to go through all of that with Laricopius, because when you're still going from West Coast to Eastern US, and that's, I mean, bugs and diseases don't recognize boundaries. So, anyway, Laricopius, or LN, it's, we started to field collect them in the, ever since 2001. And now we've got populations established enough here in the eastern US that we're able to get all the Laricopius we need just from our own populations, which is really cool. One of the people that really helped that the most was a fellow by the name of Dick McDonald, who retired, but was a foremost researcher in this area, and he spends a lot of his retired time going in the field collecting LN beetles. And so we use a lot of the beetles that he collects. And we also have started enough populations in our area that we are now able to collect from our own area. So that's one of our successes. We actually have field insectaries now that are giving us these beetles, and they seem to be thriving, and they are cutting down the Adelgid population in those areas. So all the things we wanted to see seems to be happening with the LN beetle. So over 420,000 have been released in the eastern US since 2001. The second beetle, we also rear this, and we're one of the few labs that actually rear this one. It's SD. This is actually a native of Japan, that in the same area of Japan where the Hemlock Wooly Adelgid came from. It is one of its biggest natural predators in Japan, so we thought this would be a great candidate. Now, of course, the question in everybody's mind is typically, okay, so is this another kudzu? Is this another European starling? Is this another house sparrow? In other words, is introducing these things going to cause more problems than they solve? Great thing about both of these species is that they are specific predators. The only thing that they eat is this bug, HWA. They can't, and both of these beetles, their entire reproductive cycle surrounds these bugs. SD and LN attack two different parts of that life cycle I showed you earlier. So we kind of hit the beetle, or hit the HWA more than once. And that's why I love the multi-predator approach, because I feel that these two beetles are working in tandem to be more effective at lowering the population. And we have some evidence to show that. There's many places where we've released both of these beetles, and the populations of the adelgids are what we would like to see. We're never going to get rid of the adelgid. Impossible. You saw anything that any time where you have one female that can be responsible for 300 times 300 eggs in 1 year's period of time, what that total, 90,000? And all of the eggs are already fertile and more females. Anything that does that, you're never going to get rid of. That's just almost impossible. It's like getting rid of every air pollution molecule by picking all of the air pollution molecules out. We can try our best, and we can get a lot of stuff out of the air through scrubbers and smokestacks and stuff like that, but you're not going to totally get rid of it. And that's not even our goal. If that was our goal, that would be a ridiculous goal. It's not going to happen. But what we want to do is to lower the populations in the field to establish an equilibrium between these predators and the adelgid so that they are keeping the population low enough that the trees have a chance to live. In other words, instead of having millions or maybe even almost billions of adelgids on trees, if we can cut that down to several 100,000, that's still better. So to us, success looks like having a little bit of adelgid on trees, but the trees are still coping and doing okay with it. And actually, one of the other reasons we want that is if we want evolution to be on our side, these trees need to be exposed to this predator, and we need to have some natural selection of trees that are able to tolerate it better than others. So those are just some ideas there.
Do the beetles also attack the egg sacs?
Yes.
They do, okay.
In fact, ST, that's its big thing. Well, and LN. Yeah, and they will both just decimate an egg sac. It's really fun to watch. If you want to see massive carnage under a microscope, these beetles, I guess they're like the cats of the insect world. I don't think they play with their prey, but they're pretty ruthless. I love my cat, but man, I'd be scared to be something small around it. And we keep our people doors because we love our birds.
Thank you.
Have you considered hybridizing with Pacific Northwest hemlocks into the eastern?
Yes, yeah. In fact, I mean, we haven't, we're not really involved with the genetics part of the research, but there's a lot of examination of the genetics of the hemlocks, including what are the specific genes that are causing that maybe western hemlocks and mountain hemlocks, which are the two species that are out in the western U.S. What do they have that the eastern hemlocks and Carolina hemlocks don't have? And are there certain chemicals, alkaloids, or other things that the tree produces that allows it to be more resistant? And of course, so there's kind of two angles. It's not only the angle of the assemblage of predators that might be in that community, and there seems to be some in the west that help keep the adelgid down, but also the genetics that allows the western hemlock and the mountain hemlock to survive with the adelgid.
Otherwise, you would have just had perfect control with that first predator.
Yeah, right.
Secondly, is the balsam woolly adelgid close enough to the hemlock adelgid that you could get some control?
Balsam woolly adelgid is, it's in the same genus as the hemlock woolly adelgid. Of course, it's even been here longer in the eastern U.S. We were having problems with balsam woolly adelgid before the hemlock woolly adelgid moved into the Appalachians, and it totally devastated populations of trees throughout the higher elevation areas, the Smokies, and Yeah, we actually have several other adelgids. We have a pine bark adelgid, which is native to this area. The pine bark adelgid doesn't really do any damage. It does minor damage to pine trees, but usually doesn't kill them outright, or at least as voraciously as HWA or balsam woolly adelgid does. And in fact, we're actually investigating the possibility of seeing if our beetles will actually eat the pine bark adelgid, which they haven't seemed to show a lot of interest in. So, yeah, these beetles seem to be pretty specific, and I'm pretty sure they're kind of that way with the balsam woolly adelgid. Now, and I'm not as familiar with the predators in the ecosystems where BW may be from, but yeah. But the genetics aspect of it is really a critical part of the picture. In fact, that's probably the long-term solution, is get resistance in the trees. And then we don't have to worry about this. That's our hope. So we may be stopgap with this as well. So these are the labs that are currently rearing beetles in the southeast. Virginia Tech is probably the oldest lab now in existence. They've been doing it for quite a long time down here in the south. Clemson used to be at the top of the list. Clemson's no longer involved with this project, so I don't have them up there anymore. University of Tennessee, Pat Parkman's lab up there. We've worked a lot with them over the years. Pat is now retired, and so there's other people working up there now. But and then Young Harris College was the third lab in this area, actually fourth, if you count Clemson. The fourth lab in this area that was developed, and we started our operation in 2005. University of Georgia was next, and pretty much they actually did some releases of beetles from elsewhere before our lab was established. But Yan Zhuo, who's the person who operates the lab there, we work very closely with her, and we kind of formed a coalition of Georgia labs. That's us, University of Georgia, and University of North Georgia with Stacy James's lab in Dahlonega. And so we every year try to strategize where in Georgia do these predators need to go. And we are also each doing different ways of monitoring to try to figure things out. Georgia Tech has kind of started to be involved. I've had phone conversations with some of Georgia Tech students that are interested in using drone technology to try to detect predator activity, which I'll mention a little bit about. This is our LN beetle. If you ever want to see them live, stop by my lab. I'll be glad to show them to you. They're my favorite. They're kind of the rock stars of the predatory beetle world. These are the ones that have really well established themselves in insectaries in our area now, which tells me a lot that they've adapted well to our adelgid populations, and they seem to, wherever they are at, reduce the levels of infestation in that area. So these we definitely want to make sure we continue to release. And this is our ST. These are two very different beetles. This is in a different beetle group. This one is actually a ladybird beetle, just like our ladybugs, except they're the size of a sesame seed. This looks really huge. It's really tiny, and totally black. And I can show you those too if you ever want to come by the lab. This was our first release at Soapstone Creek back in 2006. I think is when we did the first one. this area that I worked with to do these releases. Of course, he's been retired, but he's very involved still in a lot of different things. We used back then the ice cream containers, and you can see all those little black dots for our ST beetles in this release. We've worked with the Forest Service and Georgia Forestry Commission to come up with hemlock conservation areas, specific areas where we do releases on public lands. And of course, we got about 140 of these now. I think this is an older map, so I don't think we've got all of them. We put a few more in this area and even in the Cohuta's area. And there's also, now we have some way back in the western area like Walker County and those places like that. So that aren't on this map. But this is pretty much, we've got Hiawassee and Blairsville and Blue Ridge, and of course a lot of it's the forest area south of us and east of us, all the way to the South Carolina border, and then of course the Cohuta's and many different places. These are all areas where we feel are critical areas to keep hemlocks in because of their ecosystem impact, and these are areas that we target the most. Obviously, we got a lot of expanse, so we can't just be like Johnny Appleseed and throw them everywhere. It takes a while to rear these things, and it takes a lot of time and effort. And so now once they're in the field, they seem to do okay. But it's the beginning part that's kind of tough. So yes, first of all, Georgia is about the only state where multiple predator species have been consistently released. We had that strategy from the very beginning. Most of the other states where releases occurred were kind of single predator releases. And sometimes, you want to hit something at more than one part of its life cycle in order to knock it down. So we've had that philosophy from the beginning. We have done a lot of field recovery of both LN and ST beetles, especially in some of the HCAs, and these also have hemlock conservation areas, especially some of the ones that have what we want to see, low levels of HWA, and they don't seem to have this kind of boom bust cycle that we have if we don't have any protection. And so Dockery Lake is a big area that's been positively impacted by the beetles. Boggs Creek, this has turned into a really neat area because it's not very far from here to drive down there. Yeah. And we've been able to field collect beetles in those areas to then breed them in the lab again and release them. Of course, they're breeding successfully in the field. Some of the beetles in Boggs Creek that we released 10 years ago have established populations. And we know that because we haven't done any releases since then.
How do you actually collect them out there, as little dots?
It's hard.
A really fine net.
Yeah, well, kind of. It's beat sheeting is our main way of doing it. Beat sheeting is what it kind of sounds like. You take a big bed sheet, wrap it around branches of hemlock trees, take a broom handle. It's really high-tech stuff, and then you beat the branches, and then we have our little aspirators, which are these just bug suckers. And we just lay it on the ground, and then we just suck up every LN. I have two. I'll have one for LNs and one for STs. Of course, sometimes I get confused, and so I have to sort them out in the lab.
And might there be both on the same branch?
It begs the question, do these beetles have anything that prey on them, and how are they responding to predation?
They most certainly do. I mean, I don't have a ton of evidence for that except in the lab. Because in the lab, like I said, we'll take foliage from the field, we'll bring it in, and then we'll that foliage in the field, that we use to feed our beetles, that's infested, will have other eggs of other things. And things like lacewings, will prey on our beetles and the beetle larvae. Probably spiders. There's once or twice I decided to leave spiders in the cage just to see what the final result would be, and it appeared that they were preying as well. So lacewings, spiders, also the Harmonia ladybug. More so the larva of that ladybug. So the larva of the Harmonia beetle, which is the one that's often been used for control of aphids, it apparently loves our beetles. So yes, we've had to worry a bit. We aren't sure about birds. Quite likely some birds will eat them. But you know, we just don't have enough like visual evidence or field evidence, and obviously we don't have any birds in the lab so to feed on them. So, but in the insect, yes, definitely lacewings, Harmonia beetles, for sure. And my student that was working on predation, just an experiment over this past year, found some larvae that we can't identify that appear to be eating. So we're trying to see if we can find more of that particular larva so we can actually do some identification. Unfortunately, I'm a botanist and not as much of an insect person, but I have a couple of students that are just very good at insect identification. So I like to throw things at them when I find something. So we've seen some recovery 10 years post-release. We've also recovered beetles 1.5 miles from their initial release site. That tells us that they're spreading. And a lot of this has come from the research at the University of North Georgia that Stacy James, and before her, Sarah Jones, who was the lab manager there, was doing. And so, and then in our lab, we've been doing a few things too with some of the research. But we have observed less mortality and canopy loss in some areas. We've determined that through actually some satellite data in addition to field observations. I had one student last year, his name is Garen Brown, or 2 years ago, I'm sorry, who's now in a PhD program at the University of Iowa. He did a lot of field reconnaissance of some of our more remote beetle release areas around Davidson Creek, Boggs Creek, and Wolfpen Gap, and he actually was able to measure the canopy of the trees using satellite data. And I did nothing. I will tell you that on that research project. I was just there to hear what he was coming up with and what he was doing. That's a little bit beyond what my abilities are, but Garen's a tremendous whiz at GIS and all of the other technologies associated with satellite imagery. And I just provided him with the resources and said, okay, you know what you're doing. And it turned out he did. Other people recognized that. So we hope to publish that information soon, but he's pretty busy right now in his doctoral program at Iowa. This is something we've worked on most recently and had a couple of students work on this. A technique that Dick McDonald developed back in 2014 uses UV light to actually detect predation. When when the Adelgid eggs are torn apart in the predation, some of the products that are produced actually biofluoresce in the presence of UVA light. And so in the lab, we've been doing experiments on being able to detect predation in the field, and that'll give us another way for us to see how the predators are doing in the field. I had one of my students, Daniel Zimney, a lacrosse player, who was here a couple of years ago. He compared, for instance, the feeding of the LN beetle with the ST beetle, and he found out certain times of year the LN beetle feeds better than the ST beetle. Certain times of the year, ST beetle feeds better than the LN beetle. And so, which is something we needed to see. That shows us that there's different parts of the life cycle that are being hidden on. So that was pretty valuable information for us. And then Aniba Reddick, current student of mine, about ready to graduate, has actually done the same thing. We actually wondered if male or female ST beetles fed more. We figured females, you know, they lay eggs, so they need more resources, but our data actually showed that they really fed about at the same rate. So males are, you know, use as much resources as the females do. So, and you know, that's a little bit more of an esoteric thing, a little bit less practical, but still, it does help us understand these predators better. And anything that does that helps us in the battle. So, this is part of Anita's work, but you can see those little glow dots there, there, and there. That's where a beetle ate an adelgid. So there are a couple things on the horizon. Silver flies, Leucotaxis is being investigated. Leucotaxis is maybe very effective in some of the preliminary information. Of course, there's still a lot of work. It's still in the experimental stage. In fact, our lab's not involved with this at all. The Forest Service only wants a couple of labs involved with this, just for, you know, making sure that we totally understand the impact of this fly on the ecosystem before we do anything. But it's a possible another part of the arsenal. Other beetle species, there are other predatory beetles that we found in China and Japan. We're investigating. One of them was that beetle we did discontinue that I told you about. We thought it was more specific when it wasn't, so we decided to end that. But there are still other beetle species that we might get involved with. Fungi that eat bugs. There's actually several of these, kind of related to the ones that creates ant zombies. You probably have heard of. There's a few claviceps, I think is the name of that fungus. And
Cordyceps.
oh, cordyceps. I'm sorry. Yeah. And so that's something that's being looked at as well. The field monitoring techniques that we're using right now and the satellite imagery, some of the stuff that my students have been working with. If we can monitor things better in the field, we also can then target better where to put beetles, places that have more need than others. And then, like we mentioned very early on, genetic resistance. We're not involved with that. We don't really have the facilities at Young Harris to do as much genetic research, but we can do some basic stuff like, you know, PCR and some of that kind of thing, but it's pretty hard to, it's not at the level that a lot of the other university labs have. So there's a couple of Forest Service folks that are working with various universities, and I've been in communication with them to give them material to do genetic research, but, you know, they're doing the stuff, not me. So that's why I think there's hope. A few years ago, I would give this talk and I kind of would leave everybody depressed. But that's not the case anymore. I've seen so much in the past couple of years of, wow, things look like trees are surviving at a lot of these release sites. You know, so keeping my fingers crossed that it just gets better. That's all I've got.
The chemicals used to treat the trees, does it have any adverse effect on the beetles?
Yes. It does, that's why they're even, you know, I recognize we need those chemicals at certain levels. It's also, the beetles and the chemical can coexist, but you gotta strategize how to do that. For instance, it would affect the beetles. If for instance, you got an infested, or a group of infested trees, and you have put imidacloprid there, or whatever, the chemical, and then, and if there were already beetles there, those beetles would probably be affected. They would eat the egg sacs on those trees that have been treated, and yeah, they die from it. I've actually had things happen in the lab where we collected foliage from and we used a gator, and that gator had been used previously for chemical treatment, wasn't cleaned properly, and so we throw the foliage in the back, and then we feed our beetles that, and nasty death. So we had a lot of that happen, or not a lot of it, but enough to really, when it, when the amount of effort and everything we put into each of these beetles, when you
Don't
lose them
want poison.
in an incident like that. So, however, if you've done a chemical treatment when there's no beetles already in the area, and it knocks down the egg sacs, then you could introduce beetles on other hemlock trees, and they wouldn't be eating infested hemlock because, they wouldn't be eating the treated trees because they would no longer have the egg sacs. So it's a timing thing.
At Lake Winfield Scott, at the campground, a lot of the trees are treated,
Yes.
and hemlock trees are one of the sole sources of fuel wood, the dead branches.
Yeah.
And so, if you have a wood-burning stove that you take on a trip with you, Sierra brand, for instance, you cook your food over branches from a treated tree, what risks are there to campers who would be sorely tempted to
Right.
cook with hemlock branches?
That's a good question. There's probably a few kind of not terribly good answers, but there's a few things that could play into that scenario. One of them is it kind of depends on the wood. Hemlock trees will kind of partition. They'll send things that they take up systemically to the parts of the tree that are healthiest. So, a lot of the wood, if it's scrap wood that kind of fell off maybe from the tree self-pruning and that kind of thing, quite likely there's not going to be much in the clover in that. And so that's one thing. The other thing is that neonicotinoids haven't been shown to have a lot of toxicity in people, but it does in other insects. And so even if there was a little bit in there, it's possible that, and I keep using word possible because we all know that sometimes we find out later that, something has toxicity that we don't necessarily know that it has now. So I guess I probably wouldn't worry as much about it if I was camping and picking up scrap wood. But I might worry about it if I had cut down a tree and was burning wood that was a little fresher. So, and as far as toxicity on humans, that's kind of the other thing that's, most of the literature says that it doesn't seem to have as much human toxicity. We do know it has insect toxicity. We've actually, there's research that shows that. Like when seed drills are used with seeds that have been coated with imidacloprid, which sometimes happens to provide systemic protection when they plant certain kinds of crops, that's one of the goals. You know, if there's good established insect control. Now, there are some places where there will be targeted trees that have chemical control. There's also been work that has shown you don't need to treat every tree in a forest chemically. That if you treat a few target trees, that actually helps lower the spread. So it's kind of like, in a way, it's a little bit like herd immunity among people. If you've got a certain percentage of people that are immune to a particular disease, it tends to lower the spread even among the population that has, you know, that is susceptible to that disease. That strategy is actually kind of what we use for, in some cases, when we mix chemical control with beetle control in a particular area. So, one place that I'm thinking of that really highlights that in our area is Rock Creek. Rock Creek kind of down, you know, not far from the fish hatchery. What was that? Fannin County, Union County border area in the south. That there were some strategic trees that were chemically treated. They were allowed to be chemically treated. We didn't release beetles on them until the chemical, we were pretty sure the chemical, that the egg sacs in those trees were gone. And then beetle releases were put out. And in that area, I see very little Adelgid damage.
I guess you can't, treat trees with the younger trees with DBH below a certain point. Is that, would you treat all trees?
Well, you could treat younger trees. However, I would almost recommend, if I had small trees, for instance, I'm just speaking from my own property. If I had small hemlock trees or a hedge, something that I could reach, I'd probably be focusing more on the organic treatments. I'd probably be focusing more on horticultural oils and things like that to spray those. Yes, I know I'd have to do that frequently in order to keep the Adelgid down, but it would knock it down if you were attentive to the treatment. So I think that would be my strategy if I had really small trees. But yeah, I mean, you can treat smaller trees with the neonic chemicals. And in fact, a lot of times, like say George's hemlocks, when they sell, you know, they'll have like hemlock tree sales